Good day and welcome to the third part of our lecture series on human host and microbes interactions. And those lecture series are part of our introduction to microbiology lectures. I am Professor Teke Apalata, a specialist in clinical microbiology. So during this third part of our lecture series, we are going to discuss the clinical implications of bacterial genetics. So we will look at the genetics of the bacteria and their clinical implication in the human host. Okay, so let's try to understand a bit about bacterial genetics. Okay, so there are several unique aspects of microbial genetics that largely account for what we call genotypic and phenotypic diversity. You know, to have all that diverse microbial population, you know, whether it's from their outside, appearance that we call phenotypic appearance, or whether it's according to their genes, you know, the genotypic characters of those microorganisms that create all that diversity of microorganisms that we see. It's because of some unique aspects, unique aspects that are driven by changes in the genetic makeup of microorganisms. And that also impacts the ability of different microorganisms to cause disease. So some microorganisms are more equipped with certain enzymes, certain virulent factors more than others. You know, it's because of the genetic changes, okay? And they also cause an impact in terms of their propensity to develop resistance to virtually any antibiotics. So changes in genetics of bacteria is also mainly responsible for the fact that bacteria start developing resistance to many antibiotics. So bacteria have a very simple genetic organization. It's not very complex like eukaryotic cells. You know, they are haploid, you know, not diploid like eukaryotic cells. And they only possess a single chromosome, you know, a single chromosome that is a large molecule of DNA, you know, that you can see. So that's what we see here. You see here a bacterial cell with a chromosome, a single chromosome inside the cell, okay? But, so that the chromosome you can see there. But what is very important to understand first is the fact that that chromosome, the size of that chromosome, of that large DNA molecule, you know, it's bigger than the size of a bacterial cell. But how can you have a bigger thing, chromosome, able to fit inside a small cell? And this is done by a mechanism called supercoiling. Supercoiling of the DNA. It's because of the supercoiling of the DNA that we have that large DNA, you know, able to fit into a small bacterial cells. And there are two major enzymes that are involved in that mechanism of supercoiling of the DNA. One is what we call the topoisomerase, and the other one are the gyrase. Okay, very important to remember, because when we will study some antibiotics, especially the fluoroquinolones, we will understand how they act. It's because they are able to play a role against the activity of these enzymes that cause supercoiling. Then, as a result, the bacteria will then release the chromosome that is inside and explode. Okay, so we will see that. So this is a large DNA molecule, that's a DNA double helix that we call coil, as you can see. So the process of DNA supercoil, it starts by making this double helix DNA to this form, by those enzymes. Then after that, we have a DNA supercoil. Then slowly it tries to fit it inside a small bacterial cell. Okay. So then, what is next? Now, the DNA, the bacteria is then able to undergo what we call protein synthesis. And protein synthesis has two steps. There is what we call transcription and translation. So transcription happens when from the genomic DNA, there is a formation of a messenger RNA because of the enzyme called RNA polymerase. There is genomic DNA with the production of messenger RNA. Actually, the messenger RNA will carry the information from the genomic DNA of a specific protein to be formed. Then the messenger RNA will then instruct the transfer RNA to bring the amino acid into the ribosome for the mechanism of translation because of the formation of the new protein. Okay, that's how the protein synthesis happened. Okay, so, and for this to take place, you need the recoiling of the genomic DNA to take place. So look this, what happened. So you are in the cell, you have this chromosome that we saw at the beginning, and you have ribosome. So those are ribosomes with a different subunit, okay? So with the two subunits here, of the ribosome. So that's where the protein synthesis takes place. So if we take this in the ribosome, that's where the protein synthesis happens. You have the ribosome there, and you have the chromosome here, okay, the genomic DNA. So from here, the information, first, there will be a process of uncoiling of this DNA, genomic DNA, the process of uncoiling of these. Then from there will be separation of the double-stranded DNA. So the two strands will be separated. Then the information from the genomic DNA through the mechanism of transcription, because of the RNA polymerase, there will be a formation of the messenger RNA that will carry the information to the ribosome. And from there, the instruction will be given to the transfer RNA to bring different amino acids depending on the protein to be synthesized. So you have different amino acids that will then be organized at a specific fashion, to develop a new protein that is formed. So, and this mechanism is called translation that takes place in the ribosome. But then, how do we end up with biological diversity? Because most of the problems that we have, the infection, resistance to antibiotic, challenges that human hosts face it because of the diversity, the bacterial diversity. So bacterial diversity happens in two ways, or two broader ways. First, it can happen as a result of gene expression, because of the gene expression, and it can also happen as a result of changes in the genetic makeup. Okay, and when there are changes in the genetic makeup, it will be either there is gene acquisition or there is a gene loss. So let's try to look at this in much more details. Let's look at this. When we speak about switching on or off of a genes, it's about gene expression, okay? So gene expression means that in that genomic DNA, in that chromosome, where do we have various genes, some genes can be switched on or off. And when the genes are switched on, we speak about gene expression, they are expressed because of something that can stimulate those genes to be expressed, and we can see a specific result. Okay, so let's give a typical example. We have, for example, Staphylococcus aureus that, has a gene called MEC2A gene. Now, that MEC2A gene can mutate, it can mutate. Then when there is a mutation in the MEC2A gene, so that mutation will lead to something, being expressed. And we can then see phenotypically changes in the structure of the penicillin binding protein, of the site that can bind to the PBP. So there are changes there. And any beta-lactam that will be given will then, be resistant. So there will be resistance development to cloxacillin, for example, against Staphylococcus aureus. Okay, so the switching on and off of genes can happen, like they can also be because of expression of a specific gene. You will have enzymes like beta-lactamase produced that can hydrolyze some beta-lactam drugs. Okay, so those are examples of genetic expression that you can be able to see. And sometimes there is also a mechanism called quorum sensing. We spoke about bacterial growth curve. When you have a bacterial growth curve, when the microorganisms enter the logarithmic phase, they replicate so rapidly, then they need to sense the environment. So the gene that is able to understand the nutrients in the environment, whether the bacteria should continue replicating or not, will be switched on. That's what we call quorum sensing. Okay. Another example of switching on and off of the genes, in the first part of this lecture, we also spoke about Staphylococcus, about Salmonella typhi, having antigen O, H, and VI. So all those antigens, they switch on and off depending on the need. Let's say when Salmonella typhi feels that it's now taken by a macrophage, it will, when it's taken by a macrophage, it does not need O antigen or H antigen. So it will switch those antigen off. Then switch the VI antigen, the antigen that code for the production of the capsule. It then switches on. Then the capsule is formed, the bacteria is protected inside the macrophages. So those are, gene expression. Now, how does the gene expression happen, and how can we regulate gene expression? Okay, so this is what happened. This is a gene. You have a promoter, so you have a promoter, and you have here different genes. Okay, so you have different genes. So this is a gene that is switched off. There is nothing. No expression, nothing, because you need something for an expression to happen. Okay, so the entire thing is called an operon. Operon is made of promoter with different genes. Okay, so what happened then? The gene carries the different information about the genetic code of different things. So what happened is that when you have something like this, this is RNA polymerase that comes, it's an enzyme, it comes and stands on the top of the promoter. It's coming to stimulate it, to activate it, to induce gene expression. RNA polymerase come and attach there. And if there is a fit, it's going to be a gene expression taking place, depending on the affinity of that RNA polymerase with different promoters, there will be different levels of gene expression. Okay, so there will be different amounts of proteins that will be produced, and those can be then regulated. The amount can depend on the affinity of the promoter with RNA polymerase. Okay, so this is how the regulation of gene expression take place. But then the regulation of gene expression is done either through induction, and we have seen how it can be induced, or through derepression. It can be induction or derepression. Now, let's look at how the derepression happens. Now, let's say that you have the operon, but you have something else here on the top of the promoter, and that changes the shape of the promoter. It means that it will change its affinity with RNA polymerase, okay? So what happened with RNA polymerase comes, it cannot attach because the promoter is different. So there will be no expression. So in the lab, we can monitor, we can regulate gene expression, okay? We can try to put something on the promoter to prevent gene expression from happening. So even if the promoter comes, it cannot attach. Okay, so we can regulate that. So this is called a repressor protein. So what we put on the promoter to prevent expression, it's called a repressor protein. Okay, it's a repressor protein. So there is no expression of the gene. Now let's bring another protein. Try to put there. Create now a situation where we can have an affinity with RNA polymerase. So this is now a derepressor protein. So if we have a derepressor protein there, that will then bind with a repressor protein and remove it. So the RNA polymerase will come and remain there. We are in a situation where the expression becomes derepressed. So we have a continuous expression, continuous production of a messenger RNA that will continuously lead to the production of protein. Now, if the protein produced is maybe a beta-lactamase enzyme, that will then lead to the continuous resistance of that bacteria to different beta-lactam antibiotics that will be given. So it becomes maybe stably derepressed, so that becomes continuously resistant. So that's what we call gene expression. Now, biological diversity, we say, can be achieved as a result of gene expression, but it can also be achieved as a result of changes in the genetic makeup. Now, changes in the genetic makeup, we say it can be done through gene acquisition or gene loss. So let's look at this. Changes in the genetic makeup. If it happens from endogenous sources, it means that from the bacteria genomic DNA itself, you can have either mutations or you can have what we call jumping genes. So a gene that is a piece of a gene that is able to jump from one place to another. So they can also cause diversity. So diversity can happen as a result of mutations or as a result of the effect of jumping genes, okay? Or it can happen from exogenous sources. Exogenous sources means the gene of that bacteria can be able to acquire genes from the environment, from other microorganisms. They can acquire those genes, integrate them in their own genome, and become different. And those bacteria will behave differently, okay? So let's first look at endogenous sources, mutations. Mutation that happen can be just spontaneous mutations. Spontaneous mutations are simply what we call point mutations. Or they can be an incorrect repair. So there is a problem in the genome, in those genes. Some gene pieces are destroyed, the correction took place, but there is an incorrect repair. That's what we call the SOS system, okay, an incorrect repair. Or we can have also, we say we have jumping genes. Jumping genes, you can have a piece of a gene that we call insertion sequence that is able to move from one site to integrate another piece of a gene, okay, that is IS element. Or you can have a big piece that we call a transposon that can change the site. And it then creates that you have a bacteria that will behave differently because of that. Okay, so let's look at spontaneous mutation. Now you can see here this gene has a specific sequence of amino acid. And here you can see what we have here. Okay, you have, those are different amino acid. And if you look at that, focus your attention here in those amino acid, the way they are placed. So here, you can have a change. You see a change here. So this change specifically in that specific amino acid, it's a point mutation. So there is a mutation that only affects that specific amino acid that is either replaced by something else or a different amino acid. And here, you see, this is a spontaneous mutation. And those type of mutation usually happen naturally during bacterial replication, especially if the replication is too fast. Remember, especially the gram-negative that replicate every 20 minutes, there are small mistakes that can happen, and a point mutation might happen. So the presence of that point mutation might not be necessarily associated with the antibiotic resistance, for example, or by specific virulent factor, unless these can be expressed. If there is any expression, it might be there, it might not be relevant. So you can find it when you do molecular biology techniques. You find there is a point mutation, but whether this mutation has any clinical relevance, you need to see whether it is associated with something. So this is point mutation. So possible consequences of spontaneous mutation is there might be no change. That's what I say. It's just a silent mutation. It's not expressed, so it has no clinical value. Or it may or may not alter the stability or the function of the protein. That's what we call missense mutation. So it's a silent mutation. It can be a missense mutation, or it can lead to a premature termination of the process by the formation of stop codon. That's what we call a nonsense mutation. So there is a difference between silent mutation, missense mutation, and a nonsense mutation. Okay. So let's then look at the SOS, incorrect repair system. So this is, again, your gene with all the sequences of the amino acid. Then there is a damage in one strand. The damage in one strand can happen and the repair takes place. There is no mutation if the repair is correct. So you have a damage, a repair is correct, no mutation, okay? Then damage of both strands, so it might be, then you have a SOS system. Now it depends on the extent of the damage. If the damage is at a limited extent, the SOS system kept the memory of the sequence base of that gene. The repair can still take place without any mutation. Okay, but if there is a major destruction in the gene and the SOS system is not able to keep the memory of the base pairs of these genes, you know, they lost the memory, then we have a repair with mutation. So that's what we call an incorrect repair. And when there is an incorrect repair, it generates a new mutation. So this bacteria will now behave differently from other bacteria. So that is the reason we can have diversity of microorganisms. Diversity is because of these things. Beside the mutation, let's look at what we call the jumping genes, okay? So jumping genes are what we call insertion sequence elements. So those elements encode a gene called the transposase, so you have an insertion sequence that is able to jump out from one gene and to get inserted in another part of the chromosome on the top of another gene. Let's see how the jumping gene works. So here you have this chromosome that is there, and you have a gene here and a gene here. Now this gene has an insertion element here. So this is the gene R that code maybe for the production of beta D glycosidase, and you have a gene B that encode for also a beta D galactosidase, beta D galactosidase, a different enzyme. Those genes have different function. So what then happens? The insertion element, that is the IS element, can then, because of the transposase, the effect of the transposase, this can jump from gene B to gene R. Now it is integrated. So it moves here to gene R. Then directly it changes the original gene B and the original gene R. So the activity of this bacteria becomes very different. So the gene B now starts producing a different enzyme that was not producing before, and the gene R starts now producing also a completely different enzyme, a new enzyme. Okay, so that is the IS element, but you can also have a transposon. The transposon is the size is much more larger than of a IS element. The transposon can move, it jump, from that gene R to a different gene, to a gene B. It integrates now in a gene B and cause new genetic makeup. So transfer of DNA is happening within bacterial cell. We say that the transposon can transfer DNA from one site on the bacterial chromosome into another site or into a plasmid. Okay, instead of moving it from a chromosome to another site of the chromosome, it can also move it from the chromosome into a plasmid. That's how even the mosaic of resistance happened among bacterial pathogens. Okay, so the transfer can also occur in a pre-programmed, programmed rearrangement ways in some bacteria like Neisseria gonorrhea or Borrelia recurrentis, that is associated with the pathogenesis of these microorganisms. There are three methods of the transfer of genetic information. Three methods. Please, this is a very critical information. Three methods associated with the transfer of the genetic information. Okay, the first method is what we call conjugation. Conjugation is when the two bacterial cells are connected with something what we call a conjugation tube, and it's also called sex pilus. Sex pilus. It connects them, it's a conjugation tube. It allows genetic information to move, to jump from one cell, bacterial cell, into a bacterial cell. So that's what bacteria do to transfer resistant genes, among them. You see a gene that is found from this bacteria, make it resistant to specific antibiotic. Then the next time you find that another different bacteria starts becoming resistant to the same antibiotic because there was conjugation, of the genetic material through a sex pilus. And an example of this in a clinical setting is resistance plasmid uncoded, resistance that is encoded in the plasmid, with the production of beta-lactamase from microorganisms like Staphylococcus aureus, E. coli, and Klebsiella pneumoniae. So these R plasmid are transferred not only to cells, from different cells, but from the cells of the same species. So they are able to exchange, the resistant genes through the mechanism of conjugation. There is a second mechanism that we call transduction. Now, transduction is the transfer of the cell DNA by means of a virus that we call bacteriophage or a phage. So a bacteriophage or a phage is a virus that is able to infect a bacteria. So it will come, it will infect the bacteria, then it will carry a piece of the DNA information of that bacteria. Then will go and infect another bacteria, then leave that information to the new bacteria. Okay, so a piece of a bacterial DNA is then incorporated in the virus will carry it and into another bacteria when it will infect another bacteria. That's what we call lysogenic conversion. That is lysogenic conversion. And a clinical example of this is what we see in Corynebacterium diphtheria. Not all of them produce diphtheria toxin or Clostridium that cause botulinism because of that toxin, specific toxin, or some Vibrio cholera that produce specific toxin. It's because they have been infected. They have been undergone lysogenic conversion because a specific virus infected one cell, carries the information into a new cell. That new cell that was not producing that toxin start now producing the toxin. Okay, and the third mechanism or method of genetic transfer of genetic information is called the transformation. Transformation is then when the transfer of the DNA is done from one cell into another. So you have maybe a dying bacteria in the environment, the bacteria is dead, it releases its DNA, and that DNA is uptaken, swallowed by another bacteria. And this is often found in microorganisms like Neisseria, Haemophilus, and even Streptococcus. They uptake naked DNA from the environment to start behaving completely different from the way they used to be before. Now, self-assessment question. Corynebacterium diphtheriae causes the disease called the diphtheria by producing diphtheria toxin. The gene encoding for the toxin is integrated into the bacterial genome during a lysogenic conversion. The toxin gene was acquired by which process? So if you know the process where there is lysogenic conversion, then you will be able to answer. Is it conjugation? No. Conjugation, we say you have a sex pilus that link the two bacterial cells. Is it transduction? Is it transformation? Is it translocation, transposition? The best answer is transduction. So transduction, if you check here, we say it's where there is a lysogenic conversion, and the example was given here with diphtheria toxin. So this was the self-assessment question of the day. So with this, we complete the third part of our lecture series. Then follow us for the part 4 of our lecture series. Thank you and goodbye.